Okay, so this week we're going to talk about the reproductive system. This is a list of the learning objectives. And we're going to start by going through the process of meiosis. So what is a gamete? A gamete is an egg, also called ova, or sperm, also called spermatozoa. Gametes are haploid, which means they only have one copy of each chromosome. While pretty much every other cell in the body is diploid. Diploid means that they have two copies of each chromosome. One copy came from their mom, one copy came from their dad. The autosomes are chromosome pairs 1 through 22, and the sex chromosomes are the last pair of chromosomes. X or Y. XX is female. XY is male. The presence of the Y chromosome defines a male biologically. So if they have a Y chromosome, they will grow a penis and be able to produce sperm and play that role in the reproductive cycle. While if they don't have a Y chromosome, they will develop a vagina and grow a uterus, and they will be able to grow the baby inside of their uterus. So they will play that role in the process of reproduction. So again, diploid means that you have two copies of each chromosome. So when a sperm combines with an egg, the sperm carries 23 chromosomes, the egg carries 23 chromosomes, and when they produce the baby has 46 chromosomes in each one of its cells. We talked about the process of mitosis earlier on in the semester. So in mitosis, we would duplicate this number and then break it back in half so that you get equal division of the cells, so that you end with two identical copies. You start with 46 chromosomes, one cell with 46 chromosomes. You end with two cells with 46 chromosomes. But in the process of meiosis, you're actually going from a diploid cell down to a haploid cell, because what you want to do is produce the sperm or the egg in order to reproduce. So the chromosomes of a homologous pair look alike and carry the same sequence of genes for the same traits. So this region carries similar information on this chromosome pair. It doesn't have to be exact forms of the genes, though. Members of a homologous pair may carry different alleles, and alleles are different forms of the genes. So let's give an example. Let's say that your mom has brown eyes. So if we are saying that there's a location on a chromosome that codes for eye color, mom has the brown allele, and your dad has blue eyes. And let's say that location is the blue allele. As their offspring, you inherit both of these copies of the gene. You inherit two copies of each chromosome. You inherit the brown copy and the blue copy. But through heredity, one of these is going to be dominant over the other. So you're going to end up with brown eyes because brown is dominant over the blue-eyed allele. So this is just a little bit of background on heredity and such, but heredity is a topic that you're going to cover in more detail once you get to advanced A&P. So meiosis. Start talking about the process of meiosis, and I'm going to draw it out a little bit. So we have a cell with a nucleus, and there's 46 chromosomes in here. Just like with mitosis, this is going to go through a similar type of cycle. All of its genetic material is going to get replicated in the S phase of the cell cycle. And we're going to end up with one cell that has 92 chromosomes. When we enter into meiosis, we're going to go through 2 cycles of division, and we're going to end with 4 cells with 23 chromosomes each. And we're going to go from diploid to 4 haploid cells. So starting the phases of meiosis. Meiosis goes through similar phases as mitosis. First, prophase. And since these pictures are here, I'm just going to use the pictures. So I'm going to go ahead and delete this slide. And we're going to look at the pictures here. So during prophase 1, the chromosomes are going to condense and the nuclear envelope is going to start disappearing. Spindle fibers are going to start to form, and you're going to have a process called crossing over happens. And crossing over is unique to meiosis. During crossing over, the ends of the chromosomes are going to kind of swap. So you'll have the chromosomes line up in pairs, and the lower regions of the chromosomes are going to flip-flop, such that you're going to end up with these ends down here mixed and matched. Now, crossing over happens in late prophase. Then you enter into metaphase. During metaphase, the homologous pairs are going to align in the center of the cell. This is different from metaphase in mitosis, because instead of all of them aligning down the center, they're lining up in pairs now. So homologous pairs of chromosomes are going to line down the center of the cell. During anaphase, and this is all meiosis 1, so we're calling it prophase 1, metaphase 1, anaphase 1, telophase 1. During anaphase 1, the homologous chromosomes are separated and they migrate toward opposite ends of the cell. Then you have telophase 1. During telophase 1, new nuclear envelopes form. And then you have cytokinesis, where the pinching of the cytoplasm is going to separate those cells. So now we went from a cell that had 46 chromosomes, we replicated those chromosomes to 92, and by the end of meiosis 1, we're back to 46 chromosomes. But we take it one step further, and we go through a second round of this division, or this cyclic division. So we have prophase 2. Now in prophase 2, we have those two separated daughter cells. The spindles are going to form, the nuclear envelope disappears. During metaphase 2, the chromosomes align down the center of the cell. During anaphase 2, the centromeres split and sister chromatids are going to migrate toward opposite ends of the cell. During telophase 2, new nuclear envelopes are going to form. And during cytokinesis 2, these cells are going to divide their cytoplasms. So you're going to end up with 4 haploid cells. So if we compare mitosis and meiosis, meiosis occurs in germ cells, so only in the gametes, sperm, and eggs. Mitosis occurs in all body cells. Cytokinesis occurs once in mitosis, but twice in meiosis. In mitosis, the daughter cells have the same number of chromosomes as the parent cell, but in meiosis, the chromosome number is halved, so you end up with haploid daughter cells. In meiosis, homologous chromosomes pairing allows for crossing over. This process does not occur in mitosis. So meiosis is involved in spermatogenesis, genesis is the creation of something. Spermato is referring to sperm. So this is the creation of sperm. Meiosis is also involved in oogenesis. Again, genesis is the creation. Oo is referring to the egg. Spermatogenesis completes meiosis. So the sperm cells are completed by the time they are done going through the process of spermatogenesis. Now, oogenesis, meiosis actually doesn't continue and doesn't go all the way through to completion until the egg is fertilized. So the egg kind of is this pre-egg that sits on the ovary, and once a month it's stimulated to continue its division cycle and migrate toward the surface of the ovary for ovulation or for the release of the egg from the ovary. But it doesn't complete its cycle of meiosis until fertilization happens. Then and only then will meiosis be complete for egg production. We'll talk more about that when we talk about pregnancy at the end of this week's lecture. There's also some bad things that can happen during meiosis, one of them being anaphase 2 or anaphase 1 non-disjunction. If chromosomes fail to separate during anaphase 1 or 2, then abnormal gametes form. That process is called non-disjunction. So, when the chromosomes align in the center of the cell, and they migrate toward opposite ends of the cell, sometimes they don't fully split. So you end up having all 4 copies go to one side, or one go to the other side. As you continue through the cycles of meiosis, you end up missing chromosomes. This can also happen during meiosis 2. So you have the chromosomes aligned in the center of the cell, and then when the sister chromatids try to migrate toward opposite ends of the cell, you end up with both of them going to one side, while 1 and 1 go to the other side. You end up with one cell that's missing a chromosome and the other cell that has an extra chromosome. If those gametes are then used in reproduction, so during a fertilization event, it will lead to babies that have abnormal numbers of chromosomes. So here this is depicted. You can see the diploid cell, and when it divides, both copies happen to go to the one cell while the other cell didn't get any copies. So these are missing a chromosome. So if they unite with a normal sperm, that cell is only going to have one copy of the chromosome. When you only have one copy of the chromosome, that's known as a monosomy. So likely, all other 22 pairs, you're going to see the two copies of the chromosomes. It'll just be one chromosome location. So one pair is going to be missing one of the chromosomes. If you use this cell in order to produce offspring, you're going to end up with babies that have an extra chromosome. This is known as trisomy. So trisomy in the 21st location, trisomy 21, leads to Down syndrome. The XXX syndrome or triplo X syndrome leads to mental retardation and so forth. XXY, Klinefelter syndrome. XYY is Jacobs or XYY syndrome. XO is Turner syndrome. There's other types of trisomies that live, like trisomy 13 is Patau syndrome, trisomy 18 is Edwards syndrome. But with the exception of these sex chromosome abnormalities and those few autosomal trisomies, if there's a trisomy or monosomy, that usually leads to loss of the baby. This is the leading cause of miscarriage, abnormal chromosome numbers. And they say 1 in 5 pregnancies miscarry. It's usually attributed to non-disjunction events that leads to abnormal chromosome numbers.